So all you're going to need for this second lecture is simply your amino acid table that was posted under the lectures for this. All of the other lecture notes are going to be posted after we finish going through some of the questions regarding this review of macromolecules. So as we look at that term and the focus of this lecture, macromolecules, and I bet almost everybody out there can tell me right off the bat that macro means large. So a macromolecule is a relatively large molecule. Now, not every molecule in the cell is large. For example, we know that one of the molecules that we often talk about within cells is lactic acid. Lactic acid is a small 3-carbon molecule. We could also talk about many of the intermediates of metabolism. Maybe you've heard of pyruvate. That's also a small 3-carbon molecule. So there are many, many tiny molecules within the cell, but macromolecules are phenomenal in their ability to do the work of a cell, to encode for the genetic material of a cell, to provide energy to a cell. So macromolecules get a lot of the press because of their incredible role in cellular function. Now, if we look at the types of macromolecules, we'll see some pretty familiar things there. Proteins are one type of macromolecule, and that's where we're going to begin our journey of macromolecules, is by looking at the nuances of proteins. Carbs, of course, we recognize and know them well as energy storage and energy generating molecules. Nucleic acids, DNA and RNA, and lipids. We're going to look at each one of these types, but let's start with proteins. So to begin off with, let's just think about the functions of a protein. And so at this point, if you're in lecture, your TA, Kelsey, is going to pause the vodcast, and you're going to try to list 3 protein functions, and you can talk about those a little bit amongst yourselves. If you're not in class right now and you're watching this vodcast alone, I suggest you also pause it and write down what you know to be a few protein functions. Welcome back. So let's write down some of these functions of a protein. First off, we know that they can function as enzymes. That is, enzymes catalyze reactions. Enzymes allow reactions to go at a faster rate. So when we talk about proteins that catalyze reactions, these are called enzymes. Later in the semester, we're going to have a chance to talk about the nuances of enzymes, one of my absolute favorite sections, so you can be looking forward to that with breathless anticipation. The next function that I have written down, and maybe this is one of the ones that you also thought of, transport other molecules. For example, solute binding proteins. Solute binding proteins actually bind to things outside of the cell and help to bring those molecules into the cell. So solute binding proteins play a role often in transport. Proteins, as we know, also coordinate motion, and maybe one of the proteins that came to mind right away was actin. Within microorganisms, for example, within amoeba, microfilaments are actin-based proteins that can form pseudopods, which are extensions of a cell that allow for that cell to engulf certain external components. And of course, the quintessential example is the amoeba that can reach out and engulf maybe a nutrient source that happens to be outside of the cell. Proteins can also provide mechanical support. They can provide structure. We talk about microtubules and playing that role, as well as intermediate filaments inside of eukaryotic cells more often than prokaryotes. They can help control gene expression. And in fact, it turns out that proteins can literally sit upstream of genes and control whether or not those genes are turned on or turned off. We can also talk about hormones and receptors for hormones. There are peptide hormones. For example, insulin is one of those that we talked about the other day. Antibodies, also known as immunoglobulins. We'll talk more about those when we briefly cover immunology in lab. So there are very many different functions for proteins, and you may not have hit on all of these when you were listing them on your own, but these are sort of an eclectic batch of functionalities that we see amongst the protein world. Now, I have a feeling that already Candace and Courtney and maybe Britt, who's also interested in nutrition, are already thinking about the subunits that make up proteins. That is, proteins are made out of amino acids. So let's take a moment to look at what amino acids are. We recognize that there are 20 different amino acids that comprise proteins, and each one of those amino acids is made up of some unique groups. There's a carboxyl group, and those of you who have taken organic chemistry will recognize that carboxyl group. There's an amino group. There's a simple hydrogen. All are attached to the central carbon atom. And here's where things get a little more interesting. Different R groups will typify different amino acids. So we'll see a change in the R group amongst the 20 different amino acids. All amino acids will have this group, so let me circle this in a different color. All amino acids have the carboxyl, the amino, the central carbon, and the hydrogen. So that stays conserved and is found in all amino acids. On the other hand, the R group changes. So this varies amongst the 20 amino acids. And this would be a good time for you to get out your amino acid table if you haven't been looking at it and start to check that out, because I want you to notice how the R groups vary amongst the amino acids. Let's go ahead and take a look at that table of amino acids and check out all of the 20 different amino acids. So looking at this table, we can see that they all have different R groups. So let's make sure that we point that out so that everybody's on the same page. Here's that region right there that is conserved. No matter the amino acid, we see that region in all of them. So this is the conserved area that has the amino group, the carboxyl group, the hydrogen, and the central carbon. And we could just keep on doing this, right, encircling that conserved group. Which means that the place that we see the variance, I'm going to get a green pen to show that, is in this R group. So in the amino acid alanine, it has a methyl R group, a very small R group. In fact, the only R group that's smaller is in glycine, where its R group is only a hydrogen. Now we have some very large and complex R groups. For example, look at tryptophan. Tryptophan has the largest R group. In fact, I say it's so large that you might trip on it, and that's the abbreviation, tryptophan. Trip, right? So some of these are very large R groups, whereas others are very, very tiny. And in fact, it turns out that the R group is really what gives an amino acid its personality, right? We can say that there are some amino acids, because they're such small R groups, like glycine, that particular amino acid is going to be able to fit into tight spaces, right? We know people like that. It's going to fit into little areas and kinks and turns where the space is very limited. On the other hand, obviously tryptophan is not going to fit into an area like that. It's going to have a little bit more room. So as we start to talk about the personalities of amino acids, here's what I want you to do. I want you to determine which R groups are hydrophobic. Hydrophobic. That means that they hate water. Hydrophobic. So which R groups are hydrophobic? Which R groups are hydrophilic? That is, they love water. Which R groups are basic? They act as bases. Which R groups are acidic? Acting as acids. So I want you to think about that. And in fact, if you're in lecture, Kelsey will pause the podcast and you can go ahead and start to look at that. You can even split into groups, and some of you can look for those that are hydrophilic, those that are hydrophobic, those that are basic, and those that are acidic. If you're at home, I suggest that you pause the podcast and look yourself and try to determine which is which. Okay, welcome back. Let's go ahead and look and see if we can figure out which are our hydrophobic or water-hating amino acids first. In order to do that, I'm going to go ahead and erase things, and we're going to remind ourselves of what water looks like. So let's check it out. Water is going to be a molecule with 2 hydrogens and 1 oxygen. Sorry, that ruckus behind me is my 2 cats that are deciding that they need to argue right now. So if you see a cat flying by, you'll know what's going on. So here's our water molecule. We've got the 2 hydrogens and the 1 oxygen. Now, in this molecule, hydrogens and oxygens are not very good at sharing electrons equally. For those of you who speak sexy-minded chemistry, that is to say that the oxygen atom is more highly electronegative than are the hydrogens. That is, then we see that the oxygen tends to be an electron hog. It hogs the electrons more towards it at any given moment. So the oxygen will therefore have a partial negative charge on it, whereas the 2 hydrogens will have partial positive charges. That means that the water molecule overall has a dipole moment. It's an overall polar molecule. So we usually draw that dipole just like that. And hopefully everybody's remembering that from general chemistry. So water is a very polar molecule. Water-loving amino acids will also be polar, because if you remember nothing else from this vodcast, I hope you remember this: like dissolves like. So a molecule that looks like water is going to be dissolved by water. Like dissolves like. So let's see if we can find an R group that looks like water. Okay, let's scan through and see if we see anything that might be, oh, look at this. The R group of serine, it's got a hydroxyl group on it. Like dissolves like, OH, OH. So we would see that serine would be pretty amenable to water. Okay, it's not going to be water hating. It's going to be, pretty okay with water. Like dissolves like. Now, let's also remember that a molecule with a dipole moment on it is going to be very attracted to anything with a charge. So say that you have a plus charge in the area around this molecule. So maybe you've got an R group that is R positive. Well, in which case that R positive group is going to be very attracted to the negatively charged oxygen. So that particular R group is going to love water. So let's see if we can find some R groups that carry a positive charge that we're really going to love water. Oh, right here, histidine. It's got a positive charge on it, see there? So that's going to be a big water lover. Now also, if we keep scanning through, look at this, lysine, another positive charge. Lysine is going to love water, okay? How about negative charges? Well, yeah, sure, an R group with a negative charge is going to want to combine and get a little close with that positively charged hydrogen. So we're going to see then the attraction between the hydrogen and the R group. So, we'll look for a negatively charged R group, and there we have, look, aspartate loves water, as well as glutamate. There's another water lover, both of them with negative charges on them. So anything that either looks like water or has a positive or negative charge is going to love water. But on the other hand, didn't we say we wanted to know the hydrophobic or water-hating amino acids? Well, those are going to be the things that look nothing like water. They're going to be the things that look like oil. Remember how we talked about the other day that oil is hydrocarbons? So anything that's just a bunch of hydrocarbons is going to hate water. So scanning down right here where my pen's hovering, I see isoleucine. Look at that. That's going to be a water hater. This is going to be big time hydrophobic. It'll be hydrophobic because it looks like oil, and we know that oil doesn't go in water, so it's going to be big time water hater. So isoleucine is going to be a water hater. Leucine is going to be a water hater. We're going to see methionine being a water hater. All of these that have hydrocarbons in their R group, valine is a water hater. Check out phenylalanine. Oh my gosh, it's got a benzene ring. It's a big time hydrocarbon. It's a water hater. So our water haters are hydrocarbons, whereas our water lovers are either hydroxyls or they're charged molecules. Now, I'm going to erase things for a moment so we can talk about which are acidic and which are basic. Now remember, hopefully some of you are remembering some of your Gen Chem. This is like Gen Chem in 5 minutes, isn't it? Acids give up protons, right? So acids lose protons to the environment. So let's just jot that down. Acids give up protons. So if we have an R group that gives up a proton and loses that proton, so an R group gives up a proton, then that R group itself is going to become negatively charged. That is because we're looking at neutral pH, that is biological pH within our body. So at that pH, an acid will have given up its proton. So if we look and scan through, we'll find only 2 negatively charged R groups, aspartate and glutamate. Aspartate and glutamate are our 2 acidic amino acids. Now, by a like argument, we can say that bases take on protons, so they themselves will be positively charged. So check out lysine. It's totally basic with that positive charge. The other one that's even more basic is arginine. Arginine carries that positive charge, and it is base. Now I think this is pretty appropriate because I call arginine the pirate amino acid, Arg, right? So it's pretty base. Yeah, and then histidine, well now, histidine is my favorite amino acid. We could say that it's a basic amino acid, but it turns out that truly histidine is just a total proton slut. It's always giving and taking and giving and taking, giving and taking protons. So really it's a little confused as to whether or not it's basic. Sometimes it is protonated and carries a positive charge, though it is often around neutral pH, giving up and taking on protons. So we could talk about our 2 basic and our 2 acidic amino acids, and we now know our hydrophobic and our hydrophilic amino acids. And on the next page, I have a table that summarizes all of that. And you can see here that I've listed and carefully labeled those amino acids that are highly hydrophobic, those amino acids, and those have a star by them. Here are our highly water-heating amino acids. And then we have our highly water-loving amino acids with the little triangle asterisk here. Those are all of our very, very water-loving or hydrophilic amino acids, like dissolves like, remember? And then notice that all of our basic and acidic amino acids are also highly hydrophilic, right? Because they're charged. So that's a pretty cool table kind of summarizing everything. So remember how I had you take your poster project description and hang it somewhere that you're going to see a lot? I want you to take this table and do the exact same thing with it. Find that spot where you're always staring. I don't know, maybe it's the mirror above your bed or something like that. Just kidding. But wherever it is that you tend to stare a lot, put this table there and begin to learn this. In fact, on the exam, let me show you. On the exam, I'm going to give you this. So this will be attached on your exam. You don't need to memorize anything about this particular structural depiction of the amino acids. That will be on your exam. However, I will not give you this. So what that means is that you're going to have to learn the one-letter abbreviations for the amino acids. So these are actually something that you're going to have to start to memorize. And I know I promised not a lot of bulimic learning. Well, when you get to know the one-letter abbreviations, eventually it won't be bulimic because you'll use them all the time. But to start with, you just have to start by memorizing. So let's talk about some of these one-letter abbreviations. Most of them make sense, like glycine is G, alanine is A, valine is V. But when you get to some of the weird ones, well, phenylalanine is F. That's kind of confusing. So there are some confusing ones, F and Y. Y is for tyrosine. A couple of other confusing ones that we see down here, the basic amino acids, K is lysine, R is our pirate amino acid, ARG, right? That makes sense. D is aspartate, and E is glutamate, right? These are some of the ones that don't make so much sense. And in fact, there's only 7 of them that don't make as much sense that you'll have to really kind of get down and dirty with your memorization. Wonderful. Let's move on and talk just a little bit about what happens when one amino acid binds to another amino acid. So here we have an amino acid, and we can see the central carbon atom. We can see the amino group and the carboxyl group. And then finally, this kind of bright orange ball is what I'm showing as the R group. So we can see all of the conserved region is up here, and then this is the R group down here. Now we know that that can vary amongst all 20 different amino acids, and maybe it's a really large group, or maybe like this, it's very small. Maybe this is glycine and it just only has a single hydrogen atom here. So depending upon the amino acid, we see differences in that R group. Now let's look at 2 amino acids that might bind to one another. Okay, so when we look at these amino acids, we always think about it as going from the amino terminus to the carboxyl terminus, and they can bind to form a linkage between the 2. So my next question actually is, if 2 amino acids bind together, and we can see this going on here, we've got one amino acid on the left there, the second amino acid on the right. If the 2 of them bind together and form a linkage, what I want to know is, what is the name of this bond? And what molecule, question mark, is lost when this bond forms? So Kelsey again will pause the vodcast as you think about the name of that bond and what molecule is lost. And again, if you're at home, go ahead and pause it and while you think through the name of that. Okay. Excellent. So I hope everybody has thought through the name of that bond, and I'm going to go ahead and make the bond here. So I'm going to pull off the needed molecule or the needed atoms to make the bond between the 2 to form this bond. Oop! I just threw one clear across the room. So hopefully we'll get everything off of here that we need. Okay, and we form that linkage right there. And hopefully everybody said that this is called a peptide bond, and that the molecule lost is actually water. Now let's see if we can prove that. Let me scrounge up the oxygen atom that I just threw across the room, and we'll see if we can show that it's a water molecule that's lost. Okay, I got it. So we've got our oxygen, right? We've got a hydrogen here, and we've got another hydrogen here. So I have to tell you guys this story. So a while back when I was teaching, I had a really large lecture class. I was over in the classroom building, and I went up to one of my students and I said, you know, can you hold, so that I wouldn't throw the oxygen across the room, I said, can you hold these atoms as I make this bond? And I gave him each one of these, right, as I took it off, and he was holding these in his hands, and I said, okay, so what do you have there? Well, let me tell you, he was a football player and he was a wrestler. So picture him, right? Huge. And he just looks at me and he looks at what he's got in his hands and he's like, little balls. And I looked at him and I said, that's not something you might want to divulge. So little balls, not really. This is actually a water molecule that you have here. I'm elated to tell you guys that he became a teacher. So all things end well. But there you have it, our water molecule.